Okay, good afternoon, everybody. So I think we wait a couple of minutes more before to start. So hello, Koray. Your microphone.
Hola, Susana, how are you?
Fine, and you?
Good, great to see you.
Long time ago, huh?
Yes, I was thinking in showing a picture when we both were grad students or postdoc. I don't remember the title, but I decided to keep the picture for an in-person meeting in a way that it doesn't get in the recording forever.
Ah, okay. Good. I think the last time I saw you was in, I met you was in Manchester, maybe? With the,
probably.
Yes.
Yeah, back in, at University of Manchester and guessing, yeah.
Yes.
That has to be maybe 10 years now?
Yeah, maybe.
Yeah.
Yes.
Thank you so much for the invitation. It's great to be here and see you.
We are very glad that you were able to do it because we know that you are very busy now.
Yeah, I'm very happy to be around. Yeah.
You know, in Mexico, we are not so sharp in time.
Don't worry about it. And I also know that it's the afternoon talk. So I imagine that for some people, might be a long day already and they might skip. Do the people know they can? I see you are recording.
Yes.
Is this going to be available afterwards for people or?
Yes, this is the idea because, you know, there are people, they cannot stay for the whole day. For example, the people from Europe. So then we will put this, I don't know how still, if we put a link in the website or something like that, but we will let you know. So.
Yes, probably we will have a link, no?
Okay.
For all the participants of the conference.
Yeah, because now with the online thing, it's very convenient because you don't have to travel, but it's also very difficult because, for example, in 2 weeks from now, I will have a conference in Japan and it's very, very difficult. My talk will be the first one and it will be 11 p.m. for me.
Yeah.
And then, I don't know, it's, you have to be awake at 3 in the morning. There are some good things and some very bad things.
Yeah, I'm hoping that we all can see each other in person sometime sooner than later.
Yes, of course. Well, okay, I think it's time to start. So I have the pleasure to introduce you to Koray Colina. Professor Koray Colina is coming from University of Florida. She's in the chemistry department. So please, Koray.
Thank you, Susana. So it is my pleasure. Let me share my screen and make sure first that you can see it. Can you see the whole screen?
Yes.
Perfect, thank you. So it's my pleasure to be here. COVID have teach us all to try to be energetic and keep going with so many things at the same time. I want to thank Susana and all the organizers for inviting me. It's a pleasure to share the work that we have been doing in my group with polymer bioconjugates and in silico perspective. So I decided to talk about this, but before let me share what the group does. So we are called the Computational Biophysics and Soft Materials group, and we have been doing simulation work within the atomistic and the coarse grain level for many years now. So in my group, we work a lot with designing software that can help the rational design of new materials in different areas. We are known for the absorption work that we have done for polymeric membranes, polymeric materials, similar to the work of one of the speakers this morning, Vanessa, that was saying what she was able to do with the new materials. In the other area that we work in, so we work in designing these new algorithms or packages that are needed to push the envelope of the new design. But we have also been working in the other area of hydrogels as well as bioconjugates. And today,
sorry
yes?
to interrupt you. Could you put your presentation as a presentation because it's not so big? I don't know.
It's not so big. Sorry, let me just double check. Sorry for that. Let me share again to see if I did something wrong. So I'm just selecting. Is it full screen?
Yes, now it's perfect.
Okay, okay, so, thank you. Sorry for that. So today I will be talking about the work with bioconjugates that we do in the group, and we're interested there for two reasons, because we are pushing the boundaries of what can be done with polymers and proteins, not only in the wet lab or in the real materials, but also from the simulation side. We have grown as two different communities of what the protein community can do, what the polymer community can do, and then what are the challenges and how can we help move forward. So the group, the now mandatory picture, a couple of people from here have graduated, but I just want to showcase, I guess, the vibrant and great group that I have, that I'm lucky to work with. And anything that I said that is good is due to them. And if anything is bad, it's my fault. So bioconjugates, what are bioconjugates? Bioconjugates is when we have the formation of a bond between a biomolecule and another molecule. Some people think about chimeras, like when you have the animal with the head with one animal and the body of a different animal. So those are bioconjugates. One thing that I want you to keep in mind is that these bioconjugates are made from the two different molecules, but the bond could be non-covalent. In other words, something similar to a hydrogen bond, if you want to think about it, right? That is not covalent, or it could be a covalent bond. And within that, for example, I'm highlighting here the work of Tanja Weil from Mainz Institute in Germany, Max Planck Institute, where they have a beautiful review now, a few years old, where they show all what we can be doing within the chemical design of these biomaterials. So the chemical design of the polymer, how can we change and modify the polymer to be attached to a specific protein? And imagine the richness that we have within the protein work. Of course, today I won't have time to speak about all of these areas, so I decided to talk about two general ones, the ones that are widely used, the site-specific modifications of attaching a polymer chain or several polymer chains within a protein, and the multi-functional response that we can get from these biomaterials. And to highlight that, I would like to highlight since the work of two other great female researchers as well, Tanja Weil, of course, I mentioned before, but now Monica Olivera de la Cruz and Northwestern and Tin Chung in California, where they have a beautiful work of experiments and simulation working together, how the experimental work and the simulation work talk to each other. So I just want to highlight this work. It was one of the first works with the simulations. In this case, in Monica's simulations, she have non-covalent bioconjugates, and in conjunction with calculating what is called the radial distribution function of how the molecules orient, or the atoms, orienting space. So what I want to show today is what are the benefits of protein polymers conjugates. Let me start with what is known as pegylation. Many of you might have heard about it due to COVID, right? That there is one of the vaccines that some people have reactions to PEG, the polyethylene glycol in the polymer attached to the antibody. But what are the benefits of these protein polymer conjugates? What are we has been using them in the last 20 years, specifically with PEG? PEG is biocompatible, is easy to fine-tune their solubility, they have the ability to diffuse within the body, and we can target the drug delivery or the stimulus responsiveness. So polyethylene glycol or PEG is no more than this chain that you see here. We have the polymer with the carbons and an oxygen, a very flexible oxygen, but we have then the carbon we can think as hydrophobic and the oxygen as hydrophilic. And when we have the polyethylene glycol attached to a polymer, covalently attached, in this case, to a protein or enzyme, we call that pegylation. So within the studies of pegylation, I would like to highlight the work back in 2016 by Fiberivi et al., where they study the hydrodynamics radius, the effective hydrodynamics radius, in other words, how big, right, the protein polymer conjugate is. And what I want you to see here, they use BSA. BSA is bovine serum albumin. It's just a protein that you see on the left in blue here. It's around 500 residues. And they just monopegylated or multipegylated. In this case, I'm showing the monopegylated results. In other words, when they attach one polymer chain of this polyethylene glycol of around 2000 kilodaltons molecular weight or 3000 kilodalton molecular weight. So you can see the hydrodynamic radius of the protein per se is around over 2.75 nanometers. And when the PEG 2K 2000 kilodalton chain is, you can see the hydrodynamic radius increase a bit. When it goes to 3000, decrease a bit, and then when it goes to 5000, goes up again. So they said that, and then when they increase to 10,000, goes down. So they did an approximation, and they said that based on the light scattering result they have, the imaging or the light scattering results suggest that the protein in this case is in what they call the shroud-like conformation. And I apologize for my pronunciation of this, but I'm trying to pronounce shroud-like. So shroud-like is what we have on the left. In other words, when the protein is wrapped, the polymer, is wrapped to the protein or around the protein. And then they said that when they observed when the high molecular weight, actually the polymer goes all the way to the dumbbell conformation, where the dumbbell means that, as you can see here, it's like when you have a baby, right, that have the dumbbell, that la maraca, that is going away from the protein and is trying to have, when he's long enough that he makes this dumbbell or this maraca for the trying to be away from the water. So what we did later, so this is work from my group, what we did was, okay, we observed this behavior and we realized we can model this in the computer. We can do atomistic simulations with for these systems, and we can try to understand what this shroud-like or dumbbell conformation means and what type of behavior is really happening. So for that, when we need to do models, I guess that one question that I would like to ask is, when you think about a computational model, to be able to provide explanations on how the polymers interact with a protein, what is required? So I'm going to give you a couple of seconds, even though I didn't set up the poll, but just to think about if we need a computational model that is able to provide explanation on how polymer interact with a protein, which of these are needed? And I'm saying select all that apply. So I have done this poll before, and I can imagine that many of you have selected most of these answers, and that's correct. We're going to think about what the experimental polymer structure might mean, But we do need experimental protein structure that we typically can get from X-ray or NMR, but we also need what are called the force fields or the interatomic potentials that mimic the interactions that happen in our materials. And of course, we need the computational software of code that allows to do that and to be able to simulate, make the experiments in the computer, as well as they analyze and validate. So let's go a little bit in detail about that, right? So if you are from the protein community, soft materials protein community at the atomistic level, you know that we can make the molecular simulations from starting from the crystal structure or the NMR if it's a small molecule. The diffraction pattern can be obtained, the electron density map is obtained, and then we're able to obtain our atomistic model. So we have a structure where we can have, where we know what is called the secondary and the tertiary structure of those proteins. So this is already, remember, there is a huge field of protein design or protein studies. And even to study the dynamics with NMR or with simulations, we need to start having this initial structure for relatively big proteins, I will say. But if we think in the polymer side, right, we don't have that, because in the polymer, we have typically, in this case, we're thinking about protein polymer bioconjugates, in other words, in solution, in water with salt. That means that that polymer is going to be moving a lot, similar to like when you are cooking spaghetti, right? When the spaghetti is already soft and is moving a lot in the water, if you have one or two of those chains there. So which structure do we get to be able to do that analysis? So actually we don't have that. And then we end up figuring out a way of how to create in the computer, how to synthesize in the computer polymer chains that mimic what is happening in real life. And for that we first synthesize synthesize our polymer in the computer, and then we validate it against experimental data to know if we are getting results that are reasonable. So we need the structure of the protein and the polymer, and we also need to know this interatomic potential of force field. So there are several of those available, and what they take into account is we talk about what are called bonded interactions, where we take into account the bond stretching between two atoms, the angle bending, right, between atoms A, B, and C, the angle bending, or the torsional rotation, right, between atoms 1, 2, 3, and 4, using my fingers. So we have that type, we take into account that type of interaction, and we take into account the non-bonded interactions, such as van der Waals or weak interactions and electrostatics. The most important point that I want to keep here is the selection of the force field is very important, or the selection of the interatomic potential. In the protein community, we have great force fields for proteins, such as, for example, the amber force field or the charm force field. And for polymers, we also have very good force fields, such as the PCFF force field or the Trappe force field, for example. To keep in mind, though, is that now we need the simulations for those two systems together, right? That we need to be able to know that the interactions that are only for protein-protein interactions or polymer-polymer interactions, when we put now together protein and polymer interactions, we can still mimic real life. And that's true both in experiments and simulations. You can imagine from the experimental side, if you have colleagues that work in the bio area, in the medical area, and you try to take one of your polymer samples to the bio department, they typically don't let you do that, right? Because we are going to contaminate what is happening in their cell. So very similar in the simulation side. So these equations, these interatomic potentials are might be quite simple as this one, that it seems simple. But what I want you to realize from here is that we need to calculate the interactions, what are called pairwise interactions between each pair of atoms, 1 and 2, all the way to 1 to 300,000, for example, atoms that we have per time step. And we do this over and over and over in the game. So to be able to do this, we use what are called supercomputers. So let me show you results. Hopefully, if you haven't been awake or if you are joining, I'm talking about bioconjugates, and we have a protein, in this case, BSA blue in the middle, and I have the 2000 molecular weight polyethylene glycol in real size. where the oxygens are in red and the carbons are in blue. For the simulations, we did these at physiological conditions, meaning we have water, we have ions, there is pH, temperature, and pressure, and the simulations were wrong, and I'm going to show you a visualization of that. But to make easier for you the visualization, we delete the water molecules that are around here, as well as the ions, because I want you to concentrate in seeing what happened between the polymer and the protein. And moreover, I actually going to delete the carbon, and I just leave the oxygens. So again, the carbons are there, similar to what I had before. I just deleted for you to be able to track what is happening to the oxygens in this polymer. And I'm going to keep the movie running. And you can see that looks like the polymer like an area of the protein, but it's just moving a lot, right? So the polymer, in this case, polyethylene glycol likes or dislike the water to a degree. And you can see that make a slight contact with the protein. And in this case, this work was made by a graduate student, Aravinda Monasinghe, and two twins undergraduates, amazing work of Akash and Ashay Mathavan. So before I show you more results, let me just show you very quickly how do we construct the polymer. So in this case, we can be trying to mimic what is called ATRP in the chemistry of how the polymer is grown from the site in the protein all the way the polymer is, we add what is called an initiator, and then the protein start growing. So we can mimic that in the computer, and I'm just showing here the monomer for you to see. We can use what is called a pseudo-random walk to be able to grow the polymer, and every time that a new repeating unit or monomer is being added, you can see that the polymer is growing. Of course, in the computers, we use what are called periodic boundary conditions, and that's where you see the Pac-Man of the polymer. When it goes out from one side of the cell, it comes in from the other side, but then we reconstruct that for real conditions. And I'm going to stop it here because it will keep growing and growing. So let's go back to that. So we grow the polymer that way. So we grow the polymer one repeating unit at a time, one monomer at a time, and we do it from, in this case, for example, it was attached to what is called the N-terminal of the system. So what I want you to see, this is the same movie that I showed before, right? The 2K molecular weight. I'm going just to stop it. You can see that it's, it's getting right a little bit there, and it's just flying around. But in the next movie, what I want you to see now, we're tracking here where the oxygens of all those polymers, of the polymer chain, where we was moving around, where the oxygen has been. And as you can see, what is showing is that, as you can imagine, the polymer is moving a lot, and it won't allow any small molecule that would like to try to diffuse to get to this area of the protein, but all the other area is free. And using that as a comparison for the 20,000 molecular weight polyethylene glycol attached to the same site, right? So it's exactly the same initial structure that I have before, my protein with the polymer. Now the polymer is very, very, very long, 10 times longer compared with the previous one. And I'm going to do the same. I'm just tracking the oxygens and where the oxygens of that polymer chain was. And if I go around, you will see that the protein is completely covered, right? Or most of the protein is covered. So this is what we can imagine that it talks with the light scattering results that I mentioned at the beginning, right? That shroud conformation, the one that was wrapped around. And this is what we can have as an intuition from the experimental result with the hydrodynamics radius, right? How big that protein polymer was. However, let me show you now how really that polymer, how do we believe that polymer is behaving in the protein polymer conjugate. So again, this is the same polymer. I'm showing you now the long polymer chain. You are going to see changing colors in the protein. That is where there is a contact between the polymer and the protein. And we define a contact as any distance around less than 5 angstroms. If the polymer is close by a residue or an atom in the protein for less than 5 angstroms, we call that a contact, and you will see the change in color. And you can see that moreover, those residues could be positively charged, such as a lysine, for example, negatively charged, hydrophobic, or hydrophilic. Well, let's see the movie. Let's see how this looks. Look at that, right? So it's the same simulation, the same results that I showed you before. That was red all over, right? Let me stop it here for a sec, right? It was red all around the polymer, all around the protein. And we imagine that it was wrapped around, right? We imagine like the cable going around the protein. But now we see that actually that's more or less the case, right? It is around, but it actually doesn't want to be close to the protein, right? So it's flying around a lot. And you can see that there are a lot of blue contacts close to, or in other words, with positively charged residues. But those residues are in regions that we call patches that have red or green, right? Negatively charged or hydrophilic. So this is quite interesting, right? Because we have this long polymer chain that is not really wrapped around the protein, is more heavily towards one side. So this can help us a lot in the understanding of that experimental data that we mentioned at the beginning. And I'm here just showing us an example, the normalized time where we're normalizing to 1 when the polymer is in full contact with the protein for all the time of the simulation that we're analyzing. And in this case, I'm showing the top 75% up, right? So we have the top 90% type of residues, but for example, in this region, the polymer have a lot of interaction with the protein, and that region is composed of these type of residues. And here I want you to see 2 things, right? One, that we have a variety of colors because the residues have different charge and polarities. But also I would like to highlight that there are a lot of blue, right? There is a lot of, something is happening with these lysines that appears that the polymers like that. And if that's the case, and I'm going to show you a little bit more of analysis, what that means is that if we would like to design a protein where we want to highlight a polymer interaction to hide, for example, a region of the protein, we can do that relatively simply, just doing what is called a mutation, right? We change another residue for lysine, and then we force the position to be favorable, attracted to the polymer. And we can do that both experimentally and in simulations, of course. So that was a great story, hopefully, right? So it's quite different how the polymer chain looks compared long or short. But I just showed you results actually for this first plot, the 2K. Sorry, here I'm showing the residue number, in other words, the protein, as a function of time. And when the protein and the polymer have the contact between them, the 5 angstroms distance that I mentioned before. And I show you results for this movie that was the 2000 kilodaltons, the short polymer. And I show you results for the one in the bottom, the 20,000 molecular weight. What I want to highlight here is I show you results of one simulation, right? So in that simulation, the protein shape was the heart shape. So that heart shape is more or less the same always because we have the protein in pathological conditions. But the polymer chain, you saw that it moved a lot, right? So we need to be able to have good statistics of how that polymer chain is moving and to know if we're not getting trapped in what is called a local minima that might be biased based in the initial set of our simulations. So to overcome that, back a couple of years ago, what we decided to do was to start the polymer chain in a different position, arbitrary different position, and run relatively long simulations to see by that time, for the size of the protein polymer conjugates that we have, what sampling do we have? So we have 10 different simulations over here in the top for the 2000 molecular weight. And you can see that it seems that the polymer really don't want to be in contact with the protein, right? There are a couple of instances here where we have that polymer protein interaction, but in the big majority, this top area is white, indicating that there is no protein polymer interaction. If I go all the, and then if you remember that experimental plot, they have 2K, 3K, 5K, 10K, right? And it looks like it was 6 times in the size of the hydrodynamic radius that they could see. So if we move down from 2,000 to 5,000 to 10,000 to 20,000 molecular weight, you can see that even in the high molecular weight, we have some simulations, independent simulations that are showing high level of interactions. The first 4 have similar levels of interaction with residues in the 400 up and residues in the 150 down. But we have other simulations, right, where the interactions are not as strong and they are showing in these areas that we have before. This is due to the bias where how we initiate the simulations, and it is due to the length of the simulation. So from these simulations, we can, if we run them long enough, if we keep doing the experiment in the computer, we expect that we're going to see this type of results. So what we're doing is the trick of using several initial samples close to local minima that has been actually predetermined by metadynamics to be able to sample what is we believe is going, or what we think is going to happen in real life. The other point that I would like to highlight from this plot is this 10,000 molecular weight, because you will see this in the literature a lot. You can see that it appears that 10,000 seems to be like the breaking point, right? Like the critical point where something happening. Below 5,000, we have that dumbbell conformation. And above 5,000 or around 10,000, 20,000, we start having that shroud conformation. And this is true for this case of BSA PEG. I would like also to mention that we started studying this system because it's highly studied experimentally, because BSA is relatively cheap, it's easy to obtain, as well as PEG. So it's a way where we can have many different systematic studies. And let me just mention very briefly, if there is any undergraduate in the audience, we were very happy to get the cover of this, the front cover of this, especially of this issue of the Journal of Physical Chemistry B. That is just a recognition of the type of work that were done. But in this case, what I want to highlight is that the cover as well as the work, the majority was done for the 2 undergraduates, the twins that were working in this system. So that tells you how highly motivated students can, with the right resources, can really achieve amazing dreams. So let me move on. I spent a lot of time in PEG because this is widely known, is the results that we can have. We have good interatomic potentials of force field for them. We demonstrate and validate against experimental data. But what happened with all the variety of polymers, right? If we really want to design new materials for, in this case, we're doing a couple of ones, one for trying to detect, helping detect lung cancer in the early stages of life, or for brain tumors. We need other polymers. And then, for example, other polymers that are soluble are called, I'm going to call them just PAM and PINI-PAM, right? So those are 2 different polymers that are biocompatible or promisable biocompatible. What I'm showing in this slide is just to highlight the importance of those force fields that I mentioned before, right, or interatomic potentials. So for the PEG that I mentioned before, or PO, polyethylene glycol, polyethylene oxide, it's the same polymer, just depends on the size, we call it differently. What I'm showing here is just cartoons of the simulations that we obtain actually, or visualization of the simulations where we delete the waters with 2 different force fields. So what I want to highlight is this is force field A and this is force field B for polyethylene glycol, highly studied with a lot of information. We can see kind of similar behavior, right? The polymer is elongated. We can see that this force field B have a little bit of having extra interaction between the polymer, but in general, the behavior is the same. However, if I see the simulations for PAM and PINI-PAM for force fields C and D or E and F, because they are different force fields, depends on the polymer, different parameters, the striking results is that one force field said that the polymer is soluble in water, right? That's why it's elongated, it's happy in water. And the other force field says that it's hydrophobic, right? That it doesn't like the water. And that happened for PAN, for PINI-PAN, and many others. So this is key, right? Because if we select the wrong force field, and these force fields are highly used, both of them, the wrong force field in water, all of our predictions are going to be incorrect, and of course they won't be validated nor help in the experimental and the scientific knowledge that we want to improve. So what I'm trying to highlight here is that polymers in general are hydrophobic. Polymers don't like water in general. And the majority of the force field that has been done for polymers has been done for what are called polymer blends or polymers in the glassy state, in the solid-like state. So the first step is to be able to do bioconjugates for different types of polymers is to make sure that the interatomic potential that we're using are correct. We can have it with water, because if it even doesn't work with water, imagine to what can we expect for the polymer protein conjugation per se. So after doing that type of work, I just want to highlight some work that we have done with collaborators at CMU, Carnegie Mellon University, the same university that we have a speaker this morning from the chemical engineering department. In this case, it's with Chris Majaszewski in the chemistry department and Alan Russell that used to be at the chemical engineering department. And what I'm showing here is now a different protein, right? Because after we have the protocol, that's the amazing thing of the simulation work, right? And now we can test different systems. In this case, we have chymotropsin, a different protein with different polymers attached to it. So what I want you to see is that now we were able to go experimentally and in the computer to study what are called zwitterionic polymers. In other words, they have positive and negatively charged within the polymer chain, and then you can imagine as neutral, but it's called zwitterionic because the positive and negative are distributed within the chain. We can have a real neutral polymer with no charge, or we can have positively charged or negatively charged, right? And we can design these polymers, not only the length of the polymer, right? How many times we repeat the chain, or as well as the functionality as I'm showing here with different polarities. Moreover, we can do multi-functionalization. In other words, you can see here we have attached a polymer chains of the same polymer, in this case for this chymotropsin. So this work was a great challenge, but we got amazing results based in the collaboration, because in this case, Stephanie Baker was the experimental student working there. Yeah, she was able to attach the polymers in this using ATRP synthesis attached to the lysines that were in the system, in this specific system. So what I want to show you is that, so they attach the polymer. This is a cartoon made with the experimental collaborators, and they imagine all those polymer chains just going away from the protein, right? Like just like a star going away. In this case, the polymer chain, if I remember correctly, were 8 repeating units long. They really didn't want it that long because they didn't want it to wrap around, right? They just want to look like a spines, like a puerco spine, right? Like a porco spine. So when we did that model in the computer, we observed two things, right? So I want to show you the chymotropsin with the different polymer chains, right? Same type of polymer chains attached to different areas of the polymer in low concentration of salt. That's why you have just a small number of ions here, or in high concentration of salt. But let me show you the low concentration first, and let's see what we observe, right? So we have those polymer chains. You can see that they are moving around, right? They are hydrophilic, but let me stop it right there before it keeps going, right? But I don't know if you can see, but there is a polymer here that is just wrapped attached to the protein, right? So they were expecting that all of these polymer chains were going to be hydrophilic, would like to go towards the outside, but actually this polymer chain, based in the patch of the polarity of the protein, actually prefers to be, you can see that it bent over the protein, right? And that's huge information, because that will allow us to design, if we want that to happen, or it will tell us, let's don't attach the polymer in that section because it's actually going to block what we want, right? So it's not what our intuition will say. Our chemical intuition will tell us all those polymer chains are going to be pointing hours, but actually no, there is one that based in the side that is around, it just prefer to be interacting with the protein. So let me show you now the high concentration, right? Same polymers, same chains, but we now have a high concentration of ions. And what you will see here is that actually that polymer that before, that polymer chain that was before attaching to the protein, that is not happening anymore, right? With the high concentration appears that is just staying away. So this is powerful information. Let me see if I can change this slide. If you let me move on. Okay. So how can I summarize this information in a plot, right? So let me, here I'm showing you in the y-axis, the room mean square deviation. In other words, how much the polymer, the protein is moving. The protein polymer conjugate is moving as a function of time. So the first thing that I want you to see is the black line. The black line is the natural movement of chymotropsin, the protein per se. And you can see, starting from the initial structure, we are comparing how much that protein moves compared with the initial structure in water. And we can see that the protein is mobile, right? We see these dynamics of the protein, that is the black line. And then we can see what happened to the dynamics of the protein when it has the polymer chain, the A polymer chains attached, right? So let's imagine that you want to attach these polymers to the protein because you want to have that protein to seem bigger to be able to go through specific parts of the body, right? Let's imagine that you want a bigger shield because you don't want the protein to go inside the small channels that we have in the body. So if that's the purpose, we can see that adding the polymer called QA, right? We have a very similar behavior of the protein. The red line now is moving very similar to the black line, meaning that the protein keeps the same dynamics, even though it has all of those polymer chains attached. And that might be what we want, and that might be good, right? So if that's the case, if that's good, that means that green is bad, right? Because green, a different polymer, again, 8 chains together, green is saying the polymer, the protein now is very stable. It just stopped moving as much when the polymer chains are around. So those polymer chains attached in the same position made the protein less mobile. And that might not be good if we want to keep the protein with the same dynamics, for example, for what I call a step for the stability of the protein per se. And you can see in the blue right is something in between. If you want to reduce the dynamics of the protein, then green is good, right? Because green is able to reduce the dynamics of the protein. So you can see how this information can help us design and new materials with specific applications. But what other information can we have? We have a huge amount of information from, and I see my timing might be closing. So for example, like I'm showing the same 3 type of polymers that I would show before, and I'm showing the probability density as a function of the surface area that is interacting. So what it's showing is that the red protein is interacting much more, the red polymer is interacting much more with the protein compared with the blue polymer that I showed before. But moreover, these simulations allow us to have atomistic results, right? So in this case, I'm showing the index of the repeating unit of the polymer. In other words, number 1 is where the polymer is attached to the protein. So we're having a lot of interactions between the grafting site, the residue of the protein, and the polymer area. But then we see, for example, for this lysine 36, case 36, that when the protein repeating unit keeps growing, right, the number of monomers, the polymer doesn't like to be close to the protein anymore. There is no contact between any region between the protein and the polymer. So there is only contact at the beginning, and that makes sense because it's attached to it, right? However, you can see that we have other regions, right? I can pick this orange one that is quite prominent, where in this case, residue repeating unit number 6 of the polymer chain that is attached in what is called the C-terminal really have a lot of interactions with the area nearby. And that's huge. That's huge because that tells us that we can track, if we have, for example, a spin label, we can track which region of the polymer will attract, or how long the polymer can be to avoid or not that type of interaction. So we have these results, and this is just to show the difference between the two different polymers, right? The negatively charged and the positively charged attached to the same area. You can see how now, for example, the green and the polymer attached to K175 have a very different behavior than when the same with a different polymer, similar to it, but a different polymer is attached to the same site. So there is a lot of information in this analysis. So hopefully, just to try to wrap up, I'm just going to finish with this paper, because this paper is part of a collaboration, again, with Carnegie Mellon, another protein. Right now, we have lysozyme with four different polymers that we study. What I want you to see here, these are now experimental results of the stability of the protein in the presence of 100% salt saturation, where 100% means 5 molar. So when we increase the molarity, right, the salt concentration of the buffer of the medium that we have the protein in, typically the proteins are going to precipitate. And that's what we see in this black line, right? So in the black line, we have just lysozyme, and you can see that lysozyme in zero concentration have this stability, and then when we increase the concentration by 20%, by 40%, lysozyme is stable, but there is a moment where it just precipitates, right? And we're just going to have the precipitation. We can see the same thing with the lysozyme with 5 initiators. They wanted to grow 5 polymer chains from the lysozyme. The initiator is the molecule that is needed to be able to start the polymerization. And we can see that adding those initiators, nothing happened, right? It didn't affect the behavior of the protein polymer conjugate. However, when they added the degree of polymerization, in other words, the repeating units of the polymer to 18, etc., right, keep growing the number of polymers, you can see that now they have the red line, the green lines, and all the other just straight. This is incredible, because what this means is that adding more salt with the polymer, and remember that the protein can keep the dynamics, because we saw that plot before, it can withstand, it can still be stable and keep the dynamics at high level of concentrations, 5 molar, as high as the Dead Sea, right? I mean, it's just incredible. And then what they observed that with this type of polymers, right? And then when they change the polymers for the different, for the same lysozyme at the different concentrations, you can see that some type of polymers just don't work. Doesn't matter what they do compared with this type of polymers. So as you can imagine, we modeled those polymers in the computer. I'm just going to show you one result for the polymer is called PCBMA in 0 molar, the sodium chloride, or in 5 molar, right? And the initial concentrations are the pictures that you have here, just to highlight they are the polymers just pointing away. Remember, the molarity is there, meaning the ions are there and the water, they are just deleted for visualization. And what I want to show you here is this is real size scale, right? The protein in cartoon in blue, and then the polymer, the 5 polymer chains around that are relatively long, and in black are for the 0 molar. So what I want you to see is that I know that it's hard to see, but remember those plots that I showed before, the protein is still moving. You can see the polymer is moving a lot, but it's all the time extended. However, the blue one now, you saw the polymer collapse, right? So it's exactly the same polymer chain. Let me just stop the movie for a second. So it's exactly the same polymer chain, right? The 2 initial conformations. So this is the 2 initial conformations are the same. If it's in low molarity, you can see that the polymer is extended. But when we have the high molarity, that polymer collapsed. So the polymer collapsed, but it's still protecting the protein per se, and is not allowing the precipitation to happen. So with that, I'm just going to finish just saying the challenges and outlook that we need from the atomistic and the coarse grain. So I showed you results for polymer protein interactions, polymer solvent interactions, what we can get. But we do need to move on as well for the coarse grain level, because now we want to study more proteins and more polymers, right? And these simulations are relatively expensive for getting the results. So we wrote a small review a couple of years ago where we highlight those behind those details. But just want to finish saying this is just the iceberg, right? We're just in the tip of the iceberg. We're just starting to see what happened with protein polymer bioconjugates. There is a huge amount of things that we don't know. Remember, proteins and polymers are like people, right? The type of interaction that we can get is huge, but there is potential. That potential, I'm going to skip that, is related with working together experiments and simulations. And this is a review paper that we wrote a couple of years ago as well, or 3 years ago now, where we said how we can predict from the atomistic and coarse grain simulations, how it can be synthesized in this case for grafting from is called ATRP or grafting 2 with other techniques. And then how can we characterize it? And I show you just a taste of the type of results that we validate both experimentally and simulations, and what are the promising techniques that we have, with of course the idea to utilize it, right? Where we can have the applications not only in therapeutics, but also in food, cosmetics, coatings, etc. And with that, I'm going to finish thanking you for your attention, and of course the funding agencies that have helped the group over the years. And for the people in the north, this is winter in Florida. So this is February in Florida when it's relatively cold. We cannot live there, and you can see that it's winter because almost everybody have jeans. If not, they will be in shorts. And with that, I will thank you for your attention.
Thank you very much, Coral. That's wonderful talk. So now, I don't know, we have questions, please.
I see Francisco have a question.
Yes, please.
Yes, thank you for your talk, Corina. I just have a technical question, that's just for curiosity. Since I saw that you generate the polymer by just adding monomers, let's say, randomly, right? So, and you showed this movie with periodic boundary conditions in the time that so when was growing the polymer, right? So yeah. I was wondering if you take care about if this polymer is large enough, maybe you would have some problems of entanglement. So I was wondering if you take in count the entanglement that you can produce in the moment that you synthesize, let's say, in silico, the polymer.
So quite good. Then for the hydrogel work, for example, when we work in low or very similar for the high molecular weight, we are growing, and the one that I show it's a self-avoiding work. In other words, entanglements are not possible there. But with the other technique, with polymatic, that is a bit slower, we do grow the polymer. We just fill the box with all the monomers, and then we said whatever is around, make a bond. And then what we do is we minimize our equilibrate. So in that case, yes, you can have entanglements. So for the specific case of the PEG in this case, we just grow it, but you saw that the PEG want to get together, right? So it's able to make that, I won't call it entanglement, but it's able to wrap around together. But it's not going to do a knot, for example.
Yes, because well, I mean, so the polymer start to, let's say, to be compact, right? But this is not an entanglement at all.
Exactly, with the pseudo-random walk, right? Because that's one of the ways. So we synthesize the polymer that way because that's similar to what is done experimentally with ATRP, with transfer radical polymerization.
Yeah, but the problem with that is that you indeed have monomers around them. You don't have just one.
So that's why. Yeah, so for the polymer bioconjugates, this type of growing works because the polymer is going to be in a lot of water, right? So it's going to move around. However, as I mentioned, for the other work that we do with polymer membranes, for example, we use a different way of growing the polymer that is called, is a code that we developed that is called polymatic, where all the monomers are initially in the box, and then we just react all of them at the same time with whatever is close by, right? So we said if anything is between 2 or 3 angstrom or 5 angstroms, make a bond, minimize, and then do it again. So in those cases, you can get entanglements without trouble. Actually, you have to be careful because you can get ring concatenation. That is something that we don't like, for example, or we don't want.
Okay. Okay, so you take care of that. I don't know, because there are a lot of techniques about that, no? Right? Like taking some probabilities of making a bonds.
Exactly. So exactly. So that's so if you are in a system where there is high probability of entanglement, we won't grow it the way that I just showed in this movie or in this presentation. We do it this way in this presentation, in this type of work, because it's just polymer chains that are being attached, and they are not as long, I guess, to sink in the entanglement for those polymer chains.
I see. Thank you. And just taking advantage of, since I just already asked something, let me ask you something about the model of the water, because there is a lot of debate of what is the best water, the force field for the water, because
yeah. So
I come from the world of mesoscopic simulations, but when I see this full atomistic molecular dynamics, there is always a debate about the water model.
Yeah,
so
I'm there with you. And then just these simulations that I didn't go into detail. Can you see my screen?
Yes.
So these simulations, just for example, for PEG, for polymer, for the polyethylene glycol with water, doesn't matter which water model you pick, SPC, T3P, T4P, etc. The behavior is more or less the same. But if you go to this paper that I have here in the bottom, you will see there that actually we show that even the water model is quite important for, because you can have this type of behavior, having just the long polymer chain or the hydrophobic chain, just for changing the water model, keeping the same polymer model, the polymer force field. So that's huge, right? That's huge. And I don't think it's due to the water model, because the good part is that we have regions where we just want to study, in this case, it's physiological conditions, right? Or the temperature from, a storage condition that is 4 Celsius to 37 Celsius. So the breadth of the temperature is not as much, and we can play a little bit with interatomic potential or force field that we have. But yes, because most polymers don't like water, which water force field you use can affect dramatically your results. And that's why you have to be careful, and why validation is so important. So yes, I'm with you. For polyethylene glycol, again, that is a polymer that has been studied a lot. The water model with the charm force field doesn't change as much. With the amber force field, there are some issues there. And then for the other polymers, in this area that is growing, I will say rule number 1 is validation, right? Make sure that your polymer is behaving in the water model that you are selecting as it should be.
Thank you, Greg.
And for the atomistic simulations for the polymer, which force field are you using? Mainly?
So the ones that I was showing there are, we use the gas force field, that is generalized amber force field, because or the charm force field. So we're using the force field from the proteins we calculate charges using Gaussian, and calculating the charges made a huge difference for the polymer. So just picking and choosing a force field from the literature for bioconjugates, okay, it's very careful. I really want to highlight this is for bioconjugates, protein and polymers. It's very important that the force field is reasonable for the water. You take a polymer force field, PCFF, for example, or TRAPE, that are really good for polymers, and try to use it in a bioconjugate, you probably won't have good results.
Okay. Thank you. Is there any other question? Yes. Juan Donaldo, I will let you talk, I think. Are you able to talk already? Open your.
I cannot hear him if he's.
No, I suppose that I give him the permission to talk, but. Donald, ¿puedes abrir tu micrófono?
And if they want to ask me the question in Spanish, the answer will be in Venezuelan Spanish. So if I said a bad word, I didn't mean to.
Okay, go ahead.
Can you hear me?
Yes.
Of course. Well, first of all, I just want to thank you for your talk. It was very interesting. And I have rather a technical question about the process of synthesizes the polymer. You said it was like a random walk process. And I was wondering if this random walk was designed or codified by your team.
Yeah, so that's one of the things that my group, we're interested in pushing the boundaries of what the codes that are available can do. So random walk is a technique that we call it actually pseudo random walk because we grow the polymer, the monomer in each repeating unit or each monomer is attached randomly to the end. But we call it pseudo random walk because we're taking into account the force field to see the interactions, right? To see if the polymer, the next repeating unit would like to do this or would like to do that. So we attach it randomly in any position, but then we do a short, what is called minimization to allow the force field to let the repeating unit to be happy, I will call it, if you like, right? To have the right interaction and then be able to keep running. So that's one way of building. The other way that our group, and then we have Python that I didn't mention, that is, they are open source code that you can download from GitHub, for example, and you can hopefully use it. There is a tutorial manual coming on soon. The other one is called Polymatic. So in Polymatic, that is also available online and it's actually used over the world. You take that simulation box and you fill it with, let's say, 500 monomers. You said, I'm going to put randomly in the box 500 monomers. And then you are going to ask the question, who is close, which monomer is close to another one, and just make a bond. And we make a bond in the atomistic fashion, right? Meaning let's just connect it, minimize it, and see what happens. So in that one, it's not growing as the random walk. It's like when people do hypercross-linked polymers, if you know about polymers. So similar to how people grow hypercross-linked polymers, that's the way that Polymatic works.
Okay, thank you. And I got another related question. What was the difficult, or what's the difficult in trying to elucidate or know the coordinates of a polymer? For example, you said that it was very difficult and in the experimental.
Yeah, so thinking if the polymer is in solution, right? That's what I was talking today, polymer in water, thinking very thin cooked spaghetti, right? So you have the boiling water with the spaghetti already moving around and try to say, what is the structure of that spaghetti, right? So in one moment, it's like the movie you saw. In one moment, it's going to be like this, and the other moment will be like that, and the other moment like that, right? For the protein, you see a structure, you see a shape of a person, right? It's like the person. You see the structure and it's moving a little bit and you can track that. For the polymer, imagine someone dancing merengue at, 10 times faster, right? That is just how do you get that structure? You cannot get that one per se. You get an average as the one that was seen in the light scattering experiments. The simulations allow you to see that, you can stop the movie, right? It's a movie that you can stop and see how it's happening. Does that make sense?
Yeah, but I think, can you make cells of polymers or not?
Can you make what, sorry?
like protein crystals.
Yes, but if you made the protein, the equivalent of the polymer crystal, right? That is not really, it could be like a lattice, a beautiful organized polymer, like the spaghetti before cooking, for example, right? A mixture of that. But that's not the polymer in solution, right? It's like you can see the spaghetti in the box, but that's not the spaghetti after you cook it. So does it help you seeing the spaghetti in the box if you want to see cooked spaghetti?
If I get it, you try to represent the polymer in the solution. So
yeah, that is not that easy. That's exactly the issue. There is no technique, to my knowledge, that we can take average, but there is nothing where we can do what is called a spin label, right? We can put like a color in a part of the polymer and try to track where that spin label, where that specific part of the polymer is moving around. And that's something actually that we're trying to do with some collaborators to actually really show that the polymer is doing what we think is what the simulation is proposing is doing. So there are experiments called EPR, for example, that help with that. But it's not easy. If you look in the literature, you will see that there is, that's the holy grail. That's the holy grail.
Thank you.
Okay, thank you very much, Coray.
Thank you all.
Accepting our invitation, and now we move to the next speaker. So we have Jason Peña from Universidad Autónoma. No, Universidad, he's coming from Metropolitana, I think. Yes. Sorry.
You can see my screen already, right?
Yes, yes.
Okay. Well, thank you for the introduction, and thank you all for being here. As the host said, my name is Jason Peña, and I'm about to tell you how diffusion models can tell us something more about chemoreceptors, little receptors that are embedded to the cell membrane, capable of absorbing and rejecting molecules that want to go deep inside the inner structure of the cell. So let me give you a little bit of an introduction to the problem and tell you why it is important. And in order to do that, I'm going to talk to you about the importance of cells and the intercommunication process. Well, as you all may know, cells are a fundamental part of the existence of life, a little organic machine capable of making big things, thanks to which we can breathe, move, and reproduce. Not mentioning that they are actually the responsible of the embryogenesis process, the process of development. So here in your screen, you can see a lapsed video of the embryogenesis process for certain species, where cells are making almost perfect copies of themselves. But in addition to the existence of the cell, it is necessary that they work correctly, and most of the times that depends on the cellular intercommunication. This process can be reduced into 3 steps that are reception, transduction, and response. So the first step has to do with the interaction of a ligand, that's a molecule, and it might be a protein, with the receptor that's on the cell membrane. On the other hand, the transduction is the amplification of the information received by the ligand in the form of a signal. And last but not least, the response is the reaction of the cell because of the presence of the ligand. In this case, I'm showing you the DNA transcription in the cell nucleus, but of course, there are tons of types of reactions. But in this whole biochemical network, when tiny mistakes add up, this whole machinery gets corrupted. That's something that could lead into biological disorders like cancer, for example. So it is indeed a really important process. And by the scope of this research, we concentrate on the very first step of the cellular intercommunication. That is the reception and the interaction between the ligands, these molecules that are in a fluid, with the receptors. So this very first step can be modeled with the diffusion equations that are also known as the Fick's equation, the math that describes the Brownian motion. and were first proposed by Adolf Fick, as you can see here. And these are the two equations that I'm talking about. The first equation has to do with the flux that is going to be represented with J. Then we have the diffusion coefficient that in this whole research is going to be considered a constant, and the concentration that depends on spatial vector R and the time T. The second equation represents the variation of the concentration because of the changes in time and space. So these two equations are going to give us all the information that we need so we can say something more about the structure of the cell, about the structure of the chemoreceptors. But before I get into the math, I want to do a small experiment, a mental experiment with all of you to see what your intuition is telling you. So the experiment goes something like this. Imagine that you have two spheres, one of which has one absorbent and one reflective hemisphere, while the other one has a completely reflective surface, and in it has small circular absorbent patches that I actually draw, but they are so small that you can't actually see them. The thing with this secondary configuration is that these patches, if I consider the total area occupied by them, that is, if I add the area of the patch number 1 plus the area of the patch number 2 and so on, I end up occupying only a fraction of 1/6,200 of the whole sphere. So that's the reason why you can actually see them here. So in this first configuration, you can easily see that the absorbent area is 1/2 of the total sphere. And the question is, if I put these two spheres in a fluid that has certain concentration gradient, which configuration should absorb more molecules? And I wasn't able to do the poll, but I've made this poll before. And the answer seems pretty obvious, right? Because in the first configuration, we have a larger absorbent area. So that's the easy answer. But let me go back in time to 1977, when Berg and Purcell gave a really interesting answer to this chemoreception problem. And it turns out that both of these configurations can absorb the same. This is a really counterintuitive experiment because you actually wouldn't expect that. This is something really, really interesting. But this is what nature does. This is what the math is telling us. So these two configurations are the same. This is the Berg and Purcell answer. So they put little small patches with radius S on the cell membrane, spherical cell membrane. So of course, the radius of the patch is considered to be really, really small compared with the whole cell. So this is the answer that is equivalent to have a completely absorbent hemisphere than to have small absorbent patches uniformly distributed in the surface of a sphere, just as I just showed you. So then it comes the evolution of the model from an idea to a complex systems. And probably the most important modification to this kind of problem, to this model, is the Swansek modification that has to do with the interference effect. For example, if you have two receptors that are very, very close together, it starts kind of this competition effect. They both try to absorb the same molecules that there is in the environment. And then comes a Swansek and Szabo modification that includes the selectivity effect, because in reality, cells do not absorb everything that there is in the medium. They have to be selective. They have to get rid of those molecules that they don't like, that they are toxic for cells. So in this slide, I'm showing you the solution for this latter case. And we are interested in this case because we need to find the diffusion current constant that's going to tell us how many molecules can we absorb per unit time. And you have to see here, you have to realize that this solution depends on many things, but it is important to see that it uses the Smoluchowski constant, it uses the Hill constant, of course, the number of patches that we have in the surface. This K without any subscript is related with the information of selectivity, and sigma that has to do with the interference effect. And the Smoluchowski constant and the Hill constant depend on the radius of the cell, that's A, and on the radius of the patch, that is S. So that's going to be very important later. So this is the solution. This is the solution that considers the interference effect and the selectivity process in the chemoreception problem. But what about geometry? Is there any special geometry that allows to absorb more molecules in an ever small area? That's the question that we want to answer in this research. And in order to do that, what we did was a comparison between the analytically calculated constant for an elliptical receptor, where we use these two equations, and where A1 is the semi-major axis and epsilon is the eccentricity. We compare that solution with a purely geometric formulation that is right here and depends on the capital A, that is the area, capital P, the perimeter, and nu, that's a numerical parameter that's easy to calculate. So by doing this computation, by doing this comparison between these two diffusion current constant, we found this equation here that has a really special structure and that actually matches what Dudko et al. Found back in 2004. So this result is really, really important for the research. Then we extrapolate the results so you can use them in sphere. Remember that what we want is to see what happens in the very first step of the cellular intercommunication process, that is the reception of ligands and molecules. Okay, so this is the equation that describes that process. But it is time to see what happens with elliptical receptors. And because I want to do that, I get rid of the interference effect, because remember that when we did the mental experiment, what we found is that there are, we need patches so we can reach actually the absorbent hemisphere, but we don't need the whole sphere to be covered by these patches. We need only a really small area so we can reach the absorbent hemisphere. So if we get rid of that interference effect proposed by Swansig, we end up with this equation that I'm showing you here. This is our proposal for describing the chemoreception problem. So let me give you some numbers. And if we use a permeability of 50%, then the number of patches needed to reach the absorbent hemisphere is 31,416 when we are using circular receptors, and 31,337 when we are using elliptical receptors. And as a translation, that means that I'm occupying only 0.25% more area if I use elliptical receptors. In other words, 0.25% more space relative to the circular patches. And that relation is good. I mean, the price that we have to pay in this absorption and area that's being occupied in the sphere, it's really good if we use elliptical receptors. But at the end, I am using more area on the surface. So that's not what I want. What I want is to have the same area and to be able to absorb even more particles. So if I fix the area of these patches, I mean, if I fix the area of the circle equals the area of the elliptical patches, then I find this equation. And this equation is telling us something really, really special. You have to look that it depends only on the eccentricity. And that number that I'm presenting you here is always greater than 1. Math is telling us that a cell prefers to have elliptical receptors rather than circular receptors. So you can also check that if you do the eccentricity 0, that will be the same as making these ellipse to become a circle. You get a result of 1 that it matches with the physical intuition. So indeed, it is better for a cell to have elliptical receptors rather than circular receptors. As a conclusion, we can say first that it is equivalent to have an absorbent hemisphere than having little small circular patches well distributed in the sphere. And that result can be better if we use elliptical receptors. This is an undisputed improvement to the Berg and Purcell model. And of course, we have tons of applications in bioengineering, for example, to make medicines. So we still have to do the Brownian dynamic simulations, but we have a clue about the structure of the cell, and that is elliptical chemoreceptors a key to the an effective absorption. So that is everything that I have for you for my talk. And if you have any question, is well received. Thank you.
Thank you very much for your very, very good talk. So now we have time for some questions. Yes, please, Francisco.
Hi, Jason. Thank you for your talk. It was really interesting talk. And I was wondering about the last thing that you mentioned about that you are planning to make some Brownian simulations. So I imagine that your system will be like the cell, and you will model the what you are think to model as Brownian particle. I mean the motor cells, the mitochondria. What is the model that you are thinking about it?
Yes, so the model that we are thinking about is the cell itself. I mean, what we are doing here is to like grab a sphere that's going to be modeling a cell, put a little some patches with certain geometry on it, and we want to see which geometry absorbs the best. So something that I missed, that I mentioned to all of you, is that in the research we used several geometries. We used square patches, triangular patches, hexagonal patches, and n-side polygon patches, and all of them mathematically show that are less effective than if we use circular patches. And if we use elliptical patches, we have a better result. So in the simulations, what we want to achieve is to see if this is a good result, and if considering an eccentricity to be small, have any implications in the geometry of the patch. So that will be, if the eccentricity is really, really small, then it's kind of a circle. So we want to see that in the simulation. What we expect to do is to see what kind of the geometry absorbs the best.
I see. So the geometry of the cell.
The geometry of the patch. The cell is going to be always a spherical cell.
And you put patches on the surface of, I mean, on the membrane, right?
Yes,
so
that's correct.
These patches will represent like the proteins or something like that.
The patches represent the chemoreceptors, that are these receptors that are embedded to the cell membrane that have interaction.
They are proteins, right?
Yes, they are proteins, I think.
I see. But yeah, it's quite difficult, right? Because depending on which kind of cells you are planning to simulate, you will have whether a single cell surrounded by a fluid. Therefore, you will have an important parameter that you need to take in account, that is hydrodynamics interactions. Whereas if you have cells in a tissue, then you will have a lot of pressure between the cells. So I know it's, for me, it's quite difficult to see the model. I don't know if you can tell us more about the system that you are thinking in mind.
Okay, so yes, indeed, it's a challenge to do these kind of simulations, but so you can have a better view of the model. Imagine that here, this is exactly what we are doing. In this figure, you can see that the absorbing area is the black one. Okay, so this is a sphere, and of course, the absorbent area is taking 1/2 of the whole surface of the sphere. On the other hand, this image is representing kind of the same thing. But the thing here is that the sphere has a completely reflective surface, and in it has small little patches that are going to absorb molecules, that are going to absorb proteins or whatever you have in the medium. So that's kind of the model. So you can see more clearly. This is what's happening.
Okay, thank you.
Thank you, Jason. Please, Coray, you have a question.
Jason, great talk, great work. So your work is reminding me the work of Sharon Glockser. She does a lot of work with patches and shapes. And then if I understood correctly, you do the shape of the patch as, cylindrical, et cetera. But to have those patches of irregular shapes, can you use like a small spheres to make, a star or at will the patch, trying to mimic, I guess, to the type of patches that I was showing to.
Okay, yeah, so that's a very interesting highlight, because let's take another look into the Dutko solution. And the thing is that this diffusion current constant depends in the same way on the area and on the perimeter. So that means if I fix the area of it, the only thing that I have to do is find the right geometry that has the largest perimeter, so I can have a better absorption effectiveness. And it turns out that among these geometries that I just told you about, the ellipse is the one that has the larger one. But of course, we could do something to reach these more complicated geometries with different and more complex perimeters. So of course, we could do that kind of work, but it's interesting to see also that if we use elliptical receptors, we could also have a parameter that in this case will be the eccentricity, that acts as this thing that's going to tell us how much of a circle our ellipse is. And that's a great, that's a useful tool, because it's going to tell us also something more about the nature of chemoreceptors, because in nature, what happens is that chemoreceptors actually changes its shape so we can bind correctly many different kinds of proteins.
Yeah, exactly. I think that's another great idea. Asphericity, for sure, that you first play with the ratio and see what it is. And I will just suggest that maybe you can get some inspiration from Charlotte Glosser's work, because it might help design that, at the next level to the modeling that you are doing. But a beautiful work.
Thank you for the recommendation.
Thank you, Koray. So well, I think that's everything. So there are no more questions. So I want to thank you again for today. And tomorrow we will start again, I think at 9. Let me see. The program. Yes, please. Tomorrow we will start at 9. Okay, thank you for today. There is any announcement? Paco? No?
No, just that remember that the posters are online, and you will ask questions to the authors of the posters in Twitter, or maybe here during the session that is scheduled at the second session of tomorrow, right? After Francisca Guzman talk at noon, basically 12 and 10 minutes.
Okay. Okay, so thank you.
Thank you, bye.